In this lesson, we are going to discuss gene expression. At the end of this video lesson, you should be able to enumerate the flow of genetic information in the central dogma of molecular biology and explain how genes are expressed from the DNA. We already know that our DNA is our hereditary material since it consists of genes that are passed on from one generation to another. For the hereditary codes embedded in our DNA to be expressed as traits or instructions for important biological processes, our DNA will undergo two general processes for the expression of the gene. We have previously discussed the process of DNA replication, wherein the DNA makes semi-conservative copies of itself. For this genetic information to be expressed, the DNA needs to be in a different form to be received by the environment. The DNA will be transcribed to produce RNA. This RNA molecule will then be translated to specific instructions of the body in form of proteins. This flow of genetic information is what we call the central dogma of molecular biology. This important principle in biology is often stated as DNA makes RNA and RNA makes protein. Replication and transcription both happen in the cell nucleus. The RNA will then be transported to the cytoplasm of the cell where it will undergo translation. This same process is the main idea on gene expression. Gene expression is the process by which the information encoded in a gene is used to direct the assembly of a protein molecule. Gene expression starts with transcription. Transcription is the synthesis of RNA using the information in the DNA. The two nucleic acids are written in various versions of the same script, and the material is merely transcribed or rewritten from DNA to RNA. Like how a DNA strand can act as the blueprint for constructing a new additional strand during DNA replication, the DNA can also be a model for constructing a complementary RNA nucleotide sequence. The initiation of transcription involves the binding of the RNA polymerase to the promoter region of the DNA. It then unwinds the DNA strands for the synthesis of RNA nucleotides. The RNA polymerase uses the 3' to 5' DNA strand as the template strand for transcription. This is the reason why the 3' to 5' strand is called the template strand. The polymerase moves downstream, elongating the RNA transcript in a 5' to 3' direction. During elongation, the RNA polymerase still follows the rules on pairing. But for this case, adenine pairs with uracil. Remember that RNA is being synthesized here. This means that adenine bases in the DNA cannot pair with thymine in the RNA strand. However, the thymine bases in the DNA can pair with adenine in the RNA strand. In the wake of transcription, the DNA strands reform a double helix. After transcribing the gene, transcription can now be terminated. The RNA transcript is released and the polymerase detaches from the DNA. Observing closely on the two DNA strands, we can notice that the RNA strand is almost identical to the 5' to 3' strand, except for the thymine and uracil bases. This is the reason why it is called the coding strand. The RNA strand produced here is specifically called the messenger RNA. The resulting RNA molecule for a protein coding gene is a faithful transcript of the instructions for constructing the protein of the gene. It is a genetic message from the DNA through a protein synthesizing machinery of the cells that is known as a messenger of the RNA or mRNA. After having different form of the genetic information, the material can now be translated to a language that will be understood by the cells. In translation, this is the synthesis of a polypeptide using the information in the mRNA. There is a language shift at this stage. The cell must translate the nucleotide sequence of an mRNA molecule into the polypeptide amino acid sequence. Ribosomes, which are complex particles that organize the linkage of amino acids, are the locations for translation. We need to define first the terms involved in translation. During translation, the mRNA is read as a sequence of nucleotide triplets called codons. Each codon specifies an amino acid to be added to the growing polypeptide chain. The mRNA is read in the 5' to 3' direction. The next two terms are the two other RNA molecules involved in transcription. Transfer RNA or tRNA is the actual translator of the cell. tRNA acts to transfer amino acids to a growing polypeptide from the cytoplasmic amino acid pool to the ribosome. A cell manages its cytoplasm with the 20 amino acids, either in the synthesis or in the extraction of other compounds. The other RNA molecule is the ribosomal RNA or rRNA. The primary role of rRNA is in protein synthesis, in binding to messenger RNA and transfer RNA to ensure that the codon sequence of mRNA is correctly converted into protein amino acid sequences. Let us discuss the parts of the tRNA. At the upper tip of the tRNA is an attached amino acid which will bond to other amino acids during the translation process to form a polypeptide chain. At the other end of the tRNA is a nucleotide triplet called an anticodon, which base pairs with a complementary codon on mRNA. For example, if our codon in the mRNA is cytosine, cytosine, cytosine or CCC, the anticodon in the tRNA is guanine, guanine, guanine or GGG. The tRNA with a specific anticodon brings an amino acid based on the genetic code chart. A codon table can be used to translate a genetic code into an amino acid sequence. The genetic code chart is read from 3 positions. Each position is divided per nucleotide base. The left side of the chart determines the first letter or base of the codon. The upper portion of the chart determines the second letter or the base of the codon. And the right side of the chart determines the third letter or base of the codon, intersecting the first 2 portions of the table. This table will determine the amino acid that will be brought by the transfer RNA to the ribosomal translation complex. There are 20 amino acids that can be linked together in a polypeptide. Each amino acid may be abbreviated using a 3-letter or a 1-letter abbreviation. We have phenylalanine, or PHE or F, leucine, or LEU or L, isoleucine or ILE or I, methionine or MET or M. Methionine, aside from being the amino acid for AUG, is also called the start codon. Next, we have valine or VAL or V, serine or SER or S, proline or PRO or P, threonine or THR or T, alanine or ALA or A, tyrosine or TYR or Y, histidine or HIS or H, glutamine or GLN or Q, asparagine or ASN or N, lysine or LYS or K, aspartic acid or ASP or D, glutamic acid or GLU or E, cysteine or CYS or C, tryptophan or TRP or W, arginine or ARG or R, and lastly, glycine or GLY or G. All in all, there are 20 amino acids. But we have 3 codons that do not translate to an amino acid. This actually signals the termination of the translation process. The stop codons are UAA, UAG, and UGA. Let's have an example on how to use the codon chart. For example, we want to determine the amino acid for the codon CAG. We first look at the left side to look for C. For the second letter, which is A, we look for A at the upper portion. And then we look at the intersection of C from the left and A on the upper right. From there, we look at G from the right. There we can easily locate CAG in the chart. CAG translates to the amino acid glutamine. It is important to take note that we use the codons and not the anticodons when determining the amino acid. Now, let us discuss the ribosomal unit, which is a complex of rRNA molecules and proteins. The ribosomal complex is actually divided into two. The large ribosomal subunit contains the binding site for the tRNA complex. On the other hand, the small ribosomal subunit would contain the binding site for the mRNA. The large ribosomal subunit has three important sites for the elongation process. The first site is the amino acid tRNA binding site or A site. This would serve as the entry site of the tRNA. The middle site is the peptidyl tRNA binding site or P site. This will hold the growing polypeptide chain during the elongation step. This is the site which gets the amino acid from the tRNA in the A site. The last site is the exit or E site. This is where the tRNA with a discharged amino acid will exit. The flow of translation starts from the A site, then to the P site, then lastly to the E site. Now that we have discussed the terms and structures involved in translation, we can now discuss the actual translation process, starting with initiation. The first step in the initiation of translation is the binding of the small ribosomal subunit to the mRNA template. The first tRNA or initiator tRNA will now look for the start codon, which is AUG. The tRNA here has an anticodon of UAC, since the codon in the template is AUG. Following the codon chart, AUG would translate to methionine. After establishing the initiation region, the large ribosomal unit attaches to the tRNA. The tRNA is in the P site where it will wait for the other amino acids to attach to the sequence. CAG will be translated by the tRNA with the anticodon GUC. This tRNA brings the amino acid glutamine. This glutamine amino acid, which is currently in the A site, bonds with the methionine amino acid in the P site. The ribosomal complex now moves to the next codon. At this point, the first two tRNAs move to the left to accommodate the tRNA for the UGU codon. The tRNA with anticodon ACA brings the amino acid cysteine. It then connects to glutamine. The tRNA with anticodon UAC now disconnects with methionine. The complex now moves to the next codon to the right. The tRNA for AUG will now exit the complex. GUC and ACA anticodons move to the left to accommodate the tRNA for the GUC codon. The tRNA with anticodon CAG brings the amino acid valine. It then connects to cysteine. The tRNA with anticodon GUC now disconnects with glutamine. The same process will continue going to the 3' end. The tRNA with anticodon AAA will bring the amino acid phenylalanine. The tRNA with anticodon CGU will bring the amino acid alanine. The tRNA with anticodon UCG will bring the amino acid serine. Now, for the next codon, it will be UGA. If we are to look at the genetic code chart, UGA is one of the stop codons. Once the translation complex reaches a stop codon, it will signal the termination of translation. The tRNAs for the stop codons do not bring any amino acids. Thus, the polypeptide chain is released. Now, from our 24-base mRNA template, we have a sequence of 7 amino acids from 21 bases. The other 3 bases are from the stop codon. Take note that there is no 5' and 3' end for the polypeptide sequence. This amino acid sequence can now do its function in the cell. To conclude this lesson, let us review the following key points. The central dogma of molecular biology describes the flow of genetic information in cells from DNA to messenger RNA or mRNA to protein. Gene expression is the process by which the instructions in our DNA are converted into a functional product such as a protein. Transcription is the process of making an RNA copy of a gene sequence. It uses the 3' to 5' DNA strand as a template. And finally, translation is the process of translating the mRNA sequence to a sequence of amino acids during protein synthesis. And that ends our discussion on gene expression.